Our last panelist speaking will be speaking remotely. Craig Venter from the Craig Venter Institute. Um, Craig will be talking about maybe the best way to find truly alien life is to make it here. He'll also be talking about some very interesting aspects of biological teleportation. So, Craig, can you hear us?
Good afternoon. You're all right?
Yes.
Yes. Wonderful. Oh, it's even though it's very early here.
Do show up.
Do show eyes. All right, wonderful. So we decoded the first genome of a living species in 1995, and that gave us a very different view of life. I've called this digitizing biology because we're converting from the analog DNA molecule into the digital world. I'll show you later that we can go both directions. I thought it was important for context to show some of the extremes that we've seen from genome-free sequence and the life on this planet. We have organisms that at the extremes of pH, if you put your hand in these solutions, your skin would dissolve very rapidly. We have cryophiles that live at very cold temperature. The highest recorded temperature is often maybe 43 degrees centigrade. But as we go deep in the earth, we actually see with a high pressure, we actually see higher temperatures. Highest temperature recorded is 1,100 atmospheres. High salinity, Dinococcophry obdurans were from the early genome-free sequence that could take huge doses of radiations.
Craig, I'm really sorry to interrupt with a horrible echo, too. Could you make your slides full size so that we can see them better? There's a little bottom button on the right. It looks a little bit like yeah.
Did that do it?
Yes. Thank you.
Wonderful. Thank you. And also, life can survive in outer space. There's various experiments. Deinococcus radiodurans was on the outside of the space station. But life has survived for several years in space, both with radiation and just the vacuum of space. So, as I understand it, we have exchanged on the order of 100 kilograms of material between Earth and Mars. And I've been told you can't take a scoop of soil on Earth without having some Martian soil in it. We've now sequenced millions of genomes on this planet, and if I think a bigger surprise scientifically, if we find any microbial life at Mars, that it didn't match what we see on Earth. But let me tell you, life is a little bit different on Earth than a lot of people thought it was. In the oceans, people were looking for punctate organisms. In fact, when we sequence photosynthetic organisms, they're actually a cloud of tens of thousands of very closely related organisms. This is largely the same order. This is a plot of just sequences against a reference a genome, showing up to 40 or so percent sequence difference, which is pretty amazing, seeing that all mammals are less than 10% different from each other. So we see these clouds of things, in part because the two major drivers of evolution are UV light and oxygen. So these organisms are exposed to UV light, they're producing oxygen, and so we see these complex mixtures.
Now, we see the opposite if we go deep in the earth. So this is a mile deep in Colorado and a coal bed methane mine. We see huge amounts of biomass. In fact, it's been calculated there's more biomass beneath the surface of Earth than everything we can see in the visible world, all the plants and animals by weight. But we don't see the same clouds because we don't have the same drivers of evolution. And with some of the organisms we've sequenced from deep in the earth in Colorado, their closest relatives were to a volcano in Italy. So we see very different extremes depending on whether we go deep in the earth or in open oceans. But I contend DNA contains all the necessary information for cellular life. And we prove this by going the opposite direction from sequencing. We started with the DNA digital code in the computer, and we synthetically made a genome of a new species. We started early on with a virus, 5X174. We synthesized the DNA. The DNA is infective. If you just put it on the lawn of E. coli, it gets inside E. coli and starts taking over the cellular machinery, massively producing billions of copies of itself. And these clear plaques that you see are places where the virus has killed the E. coli. I call this a situation where the software is building its own hardware. We just put in DNA software and we end up with very infectious viral particles that can rapidly expand. So we went up several orders in complexity, making a million base pair genome. We tried to design a minimal cell. All of our designs failed when we made the chromosome and tried to boot it up. And it took us a long time by trial and error. It turns out a quarter of the genes that are absolutely essential for life were of unknown function. So it took a long time to get the set of those. But you take any of those genes out of a cell and you can't get life. The way we boot up a chromosome is we transplant it into another cell without a genome. This cartoon shows putting it in. We got mismatch of restriction enzymes. They chewed up the parent DNA in the cell, expressed from the synthetic DNA. And then we're left with a cell. It's the body of one species and the genetic code of another. But in a short while, we end up with an entirely new cell driven by the code that we put in it. We call this synthetic life. It's life driven by a synthetic DNA molecule, and we're currently trying to do this in cell-free systems. So life as we know it is a DNA plus RNA software system. And we've shown that if you change the software, you change the species. We've also shown that we can interconvert DNA to the digital code and going back to DNA. And this has certain implications. Several years ago, we did an experiment out in the Mojave Desert with a team from NASA Ames, including the chairman, Pete Worden. These were our spaceships. They were laboratory buses with DNA sequencers. We took samples. You can see we had very good help out there with Pete. We sequenced the genomes in the desert and sent the digital sequence back to the Venter Institute for reconstitution. So this can happen easily robotically now. And new sequencing devices are getting incredibly small. An entire automated system is being developed by Oxford Nanopore that I'll show you that will fit in the palm of your hand. But if we could sequence any DNA on Mars, we could just send the sequence back as electromagnetic wave in as little as 4.3 minutes when Earth and Mars are at their closest, which is a lot faster than rocket travel. This is the being developed Oxford nanopore system that does everything for preparing the sample and converting it into digital code. It'd be easy to send these to Mars and just have, if there's DNA there, which would be more surprising to me if there wasn't, and the under Mars oceans or from the soil, we could then try to reconstruct the Martians in secure labs in case they were different. I think we need to be looking for DNA organisms. Several experiments and tests have shown these molecules seem to be ubiquitous. I think if life is not DNA-based or RNA-based in other places, that would be a major finding. That's it from the DNA world. Thank you very much.